module, we're going to understand basic concepts and principles of how we undertake exposure analysis, specifically in the context of biological exposures, and describe the methodologies we might use for such exposure assessment. Hi, this is Dr. Meghan Davis. I direct the Johns Hopkins Poe Center, and today we're going to talk about biological exposures. Let's talk about some of the principles and what makes biological exposures different from other exposures. If we consider the different kinds of harmful agents, we might say these are different types of exposures. These can be physical, which can include noise and radiation. They can be chemical, such as pesticides or volatile organic compounds. Today, we're going to focus mostly on biological exposures. These are often microbes, so disease agents, as well as allergens and other kinds of organic exposures that come from living things. In this, we equally need to consider ergonomic, safety, social, and psychological kinds of exposures and how these might all interface together. Biological exposures are often infectious. These can include either the infectious agents themselves or products they produce. So they can be viruses. With bacteria, they can be the actual bacterial populations, but they can also be their products, such as endotoxin, which is the lipopolysaccharide coat of a gram-negative bacterium, and may persist even after that bacterium is no longer alive or viable. Some bacteria produce biological toxins, such as enterotoxins. And here I have an image of a methicillin-resistant Staphylococcus aureus, or MRSA bacterium. And this is one of the bacterial types that can produce enterotoxins. Infectious agents also include parasites, as well as fungal organisms, which can be fungal pathogens and molds. And then there are some other kinds of infectious agents, such as prions, which are an infectious protein. Biological exposures also include exposure to animals or insects or plants and their products. And so a lot of these can produce allergens, which are epitopes, such as MUS-M1, which is a mouse allergen, and that can incite different kinds of health outcomes. If we think about exposure and the toxicological paradigm, or the environmental health paradigm, as some people call it, we recognize that exposure to agents occurs along different routes of transmission or exposure, and that the relation between exposure and health effects can be modified by things like susceptibility factors. The goal of all of this is really to inform risk assessment and risk management, especially in the occupational health setting, where some goals might be to eliminate the source of the agent or reduce the exposure of a worker to these different hazards. This is true for biological exposures as it is for chemical and physical exposures. And equally, when we think about the toxicological paradigm and try to unpack the different steps between exposure and health effects, we recognize that biological exposures can equally have an internal dose. They can have a biologically effective dose, that is, where the agent is at its target. So you might think about a virus at a cell that is about to infect the cell. And then you have the early biological effects as you start to see the cellular or molecular changes, which ultimately lead to altered structure and function, for example, at an organ level, and that this is manifest in health effects like symptoms. And the various susceptibility factors and other modifiers can influence how exposure becomes a health effect. We've done some thinking about how infectious diseases fit into this paradigm. They're a little bit tricky because they tend to be more dynamic than typical toxicant exposures. For example, time or concentration in dose and host response may depend on prior exposure. So for example, if we think about a source of a toxicant that goes through food, water, air, soil, or dust and, exposes someone, that an infectious agent, you might have more dynamism. So for example, that particular agent could multiply in food. So you might have a certain inoculation dose into the food product, But by the time someone is exposed, it could be at a much higher dose than you would expect. You can also have things in the environment that can reduce the exposure. And then once in the body, as the agent is multiplying, that can really affect the kinds of things you see in terms of the infected cells and the symptoms in terms of the disease. But then there's a feedback loop where people who become infected can shed the organism and be a source of exposure, but also people who become infected might develop immunity and therefore would be resistant even if they are exposed again. And there can be some benefits if you have herd immunity. That is, when you have enough people within a population who have immunity, it might confer some protection to those even who are not carrying immunity through either vaccination or natural exposure. The other thing to consider in terms of infectious agents, as it might relate to interaction with chemicals or physical exposures, is that infectious agent exposure can drive such use of these agents. So, for example, if you have an infectious agent, you might treat someone with antibiotics. You might use disinfectants in the environment. You might expose the environment to ultraviolet light. We'll talk more about that elsewhere. And with other things like animal exposures, you might be using pesticides or pest deterrents and repellents. And so this can lead to more exposure to chemical and physical agents as a result of the infectious agent exposure. This can be challenging because you may see interaction. This could include synergistic effects. For example, if the infectious agent targets an organ system that is the same organ system that is targeted by a hazardous chemical agent, then you might see worse disease as a result of that interaction. And you can also have feedback loops where the use of the chemical could influence the infectious agent itself. Let's look at an example of a feedback loop. In this case, thinking about uses of disinfectants and antimicrobial drugs to treat pathogens could also then expose the pathogen population to these chemicals and select for more drug-resistant strains within that population. We did a study looking at chlorhexidine use on people who had MRSA infection and identified the potential for chlorhexidine resistance within the environmental MRSA that we were studying. And this was higher among those who had been randomized to use of chlorhexidine to treat their MRSA. And so this just is an example of how such chemical use can exert selective pressure such that over time, the hazardous exposure to the infectious agent could become more and more challenging to mitigate. Another thing that's important to consider with infectious agents that may be different from chemical or physical agents is that with the source of exposure, a chemical or physical agent will have its fate in transport. It will potentially be transformed within environmental media, and that can do things that might influence the hazard from that agent. With infectious agents, we think more about viability. And so here I have an example of a non-enveloped versus an enveloped virus. So non-enveloped viruses are a little bit more hardy than enveloped viruses, which have to literally butt out of the actual cell membrane and carry a little bit of that membrane around them. That means that if that membrane is disrupted by different things in the environment, including ultraviolet light or solar exposure, as well as desiccation or drying out, that they will potentially be less viable over longer periods of time. And so this can influence, for example, the infectious dose of the exposure of a worker within a system that has these viruses. Equally, the routes of exposure are particularly important. So there are many different modes of transmission of infectious agents. It can be airborne, which could be through droplet or aerosol. It can be through direct or indirect contact. Direct meaning actual physical contact, and indirect usually through an environmental medium. So a surface such as a doorknob or water or dust. It can also be vector-borne, that is carried by an insect. And then this really relates to the routes of entry, which again can be inhalation, direct contact, or dermal, ingestion, or injection. And this influences the organ systems that might be targeted or affected. An example of this is anthrax. If you have inhalational anthrax, and so we think about anthrax that might be a bioweapon, right? That this is very severe because it targets the respiratory and cardiovascular systems. However, anthrax, that is maybe a more natural exposure from use of animal hides, is a dermal exposure, skin exposure. And cutaneous anthrax is much less severe. And so the route of exposure is just as important as the agent for some kinds of outcomes. With infectious disease exposures, host susceptibility is critically important. This can refer to specific immunity, such as from prior vaccination or natural exposure, meaning that someone is immune to a particular strain or kind of infectious agent. But it can also rely on general immune status. And so, for example, as people age, they may lose some of their general immune responses, and that might make them more susceptible to certain kinds of infectious agents. Comorbidities can be important too, especially if they impact barrier functions. For example, a worker who has chronic rhinosinusitis may be more susceptible to infectious agents that target the respiratory system and upper respiratory system because the barrier functions within the nasal sinuses may be disrupted. And so it may be easier for that pathogen to gain entry and cause disease. In addition, there may be particular susceptibility windows for certain kinds of agents based on their pathophysiology. So, for example, Zika virus, if it infects someone who's pregnant in certain trimesters of pregnancy, can lead to microcephaly, which is underdeveloped brain function within the fetus. And so that's a particular susceptibility based on the developmental window. And so understanding what those exposures might be is very important. Finally, there may be behaviors that increase or decrease host susceptibility, such as use of personal protective equipment. If we come back to definitions of exposure, right? So classically, we would define exposure in environmental health or occupational health as any host contact with these chemical, physical, or biological agents. But we think also about the contact between that particular stressor, like the agent, and its receptor, whatever the target system, organ, host cell type is. And so we might have specific legal definitions that we need to think about in the context of biological exposures. One example is bloodborne pathogens. So an occupational exposure in this case would mean any reasonably anticipated skin, eye, mucous membrane, or parenteral contact with blood or other potentially infectious materials that may result from the performance of an employee's duties. And so in this, you might think about someone who is a laboratory technician who may be processing blood samples from patients that are potentially infectious, whether that processing is for understanding an infection in that particular patient or not. With our different sources of exposure with infectious agents, we need to consider not just the occupational exposures, so those occupations where there are higher risks for exposure, but also the larger context of the community environment, which could be home, could be school, it could be through other members of the family, whether there might be sources of exposure through medical treatments that someone is undergoing, and whether there are additional dietary exposures, whether these are, again, sort of accidental or deliberate. And again, coming back to the routes of exposure being critically important in terms of potential transmission, we think about inhalation as targeting the mouth or nose all the way through the lung system. And ultimately, because of the functions of the lungs, which have a very tight interface with the cardiovascular system, entry potentially into the bloodstream. We might have dermal absorption So skin contact can also lead to something more systemic. Ingestion, the same thing. Things that are ingested may stay local to the gastrointestinal tract, or they can go systemic as well. And injection, of course, into subcutaneous tissues and ultimately potentially systemic transport. And so here we have an occupational group that is highly exposed to biological exposure. So I have a farmer here. And so we might think about airborne exposures as having contact with the lungs and upper respiratory tract. Water exposures could be through the gastrointestinal tract, through ingestion, or dermal exposure of the skin. We have direct and indirect exposure to animals. So you might have direct contact with animals, through the skin. There may be opportunities for upper respiratory or even lung exposure. And then indirectly through food to the GI tract. I once did a study in the agricultural health study, and I was looking at different animal farmers and was looking also at their dietary preferences. And it was interesting because for some reason, the chicken farmers like to eat chicken. The hog farmers like to eat pork. And so you do have to think about these additional routes of exposure. I should also say, this is a definitely do as I say, not as I do moment. I had a moment when I was in training as a veterinarian and was working with cows, doing routine work with them. But we would take a break around midday and the farmer would bring us, some milk chugs and some donuts. And I will say that we were not the best in terms of our cleaning up before ingesting food. So you might definitely think about these ingestion routes for certain kinds of animal workers. We also have to recognize that as these infectious agents get into soil and dust, that they can be resuspended. And so you can have both direct contact with soil or dust, but also the resuspended particulate matter in the air that can have adherent bacteria or other infectious agents. And so you can also get respiratory exposure that way. And as I've mentioned, the route of exposure is important in terms of what it means for where it goes in the lungs and what kinds of disease you might see. And so we can have organic source gases. We can also have aerosols that are infectious. So some viruses will transmit via aerosol. And so this will be influenced again by respiratory rate, the concentration, duration of exposure. And we also think about the size fraction as being important in terms of where within the respiratory tract it might go. And so something like a Staphylococcus aureus or MRSA is more likely to be impacted on the interior nares or within the sinuses, or potentially swallowed and colonize the oropharynx than necessarily going all the way deep into the lung, whereas some viruses are definitely small enough to get down into the lung tissue. Dermal contact is really important for a lot of occupational infectious agents. And again, where the site is, what the skin condition is, and the properties of the various agent can be important. But this can include allergens, as well as infectious agents that are what we call colonizing opportunistic pathogens like Staphylococcus aureus that have little, sticky things that they grab onto skin and they'll hang out. So you can have an exposure and then later you could have an injury, and the exposure may be days or even weeks or months prior to the injury. But because of this ability to colonize and wait for an opportunity, you will sometimes see more severe disease as a result of that. Other infectious agents, such as ringworm, which is a fungal pathogen, will actually invade the skin and cause disease that way. And then there are others where the skin is a route of entry, like Lyme disease, which is caused by Borrelia burgdorferi, which is a bacterium, but it is vector-borne. And so it requires the bite of a tick in order to transmit. And so the image shown here is a classic erythema migrans or bullseye rash following a tick bite that conferred infection with Lyme disease. These can also be opportunities. In this particular example, if a bullseye rash is noticed, then it is possible to give treatment with antibiotics to prevent the the more severe sequelae that you can see with Lyme disease that can include polyarthritis and neurologic disease. Ingestion is another quite common route of exposure for biological exposures. It's less common for many of the chemicals, but it depends on the conditions of the exposure, either at the workplace or outside of the workplace. It can also be contact-based, so hand to mouth. And I'll say, again, in the personal stories department, as a veterinary student working with large animals, it is common for a certain parasite, Cryptosporidium, to be highly present, we'll just say, on dairy farms. And in particular, calves will get it and will shed in just gigantic numbers. And so the crypto will stay in the soil and in the dust. And where I was training, there can be sort of windy days where the dust gets kicked up. And after one of these windy days, I had my, inauguration with Cryptosporidium, which is a rite of passage for many large animal veterinarians, we'll just say. But I've always suspected that the route of transmission was actually dust that got on my face. And then I had an inadvertent ingestion exposure because the dust was just, on my face, and you accidentally can lick your lips and have that kind of ingestion. But this is also important for allergens, especially food allergens, as well as certain kinds of infectious agents. And so we think about GI viruses like norovirus, which is a big deal for certain kinds of people who might, for example, work on cruise ships where there's a lot of potential occupational exposure, as well as different bacteria like Campylobacter. And bacterial toxins often are the sources of food poisoning. So when Staphylococcus aureus secretes its enterotoxins, that's usually in food, and it is the ingestion of the toxin that leads to the food poisoning incident. I've already given you my Cryptosporidium parasite example. In healthcare settings especially, but in other settings as well, injection is a really important route of exposure to biological agents. And so we think about this a lot for our health-related professions like physicians, nurses, dentists, veterinarians like me, as well as all the allied health professionals. But I think what's important to remember is that your facilities and housekeeping service occupations may actually be at extremely high risk because their exposures may not be expected. For example, if they're just cleaning up the bedding of a patient and there happens to be a stray needle in there, they may inadvertently have a needle stick injury. And this is important, especially for human-to-human transmission of human-related infectious agents. With exposure, we need to consider the various dimensions. And this is true for chemical and physical agents too, but is a little trickier for infectious agents. So the duration, the length of exposure, or how long per exposure is important. So for example, if you walk into a room after someone has been in that room who is infected with measles and is shedding, the measles virus can stay in the air for hours. And so how long you're in the room and how high that dose is or exposure level can determine whether it is substantial enough to overcome perhaps immunity from vaccination. So if you have a really overwhelming dose, sometimes it can break through. And then the frequency is important too, because again, this relates to the body's ability to mount an immune response. And the higher the concentration, the more frequent or the longer duration, the more probable it will be that the infectious agent can overcome the immunity that the host is trying to mount, the worker is trying to mount. And so we need to quantify all of these dimensions in terms of the time course of the health outcome too. With a lot of our infectious agents, we think mostly about acute health effects. You get exposed to an infectious agent and then what results would be an infection. And so you're thinking about the short-term concentrations. But some biological exposures and even infectious agents may require a different dimension, right, of chronicity and different lag times. So for example, we have talked about Lyme disease as having the potential for an initial infection, but then it can also have these sequelae that may happen at a lag. They may happen months and months later, and it's hard to kind of go back and reconstruct what that exposure was. This is true for a number of other kinds of infectious agents, but then we also think about allergens and how some exposures to allergens may be the inciting or sensitizing exposure. And then you won't necessarily see a health effect in the short term from that. But with re-exposure, you may see an exacerbation. And so with this, we think about the dimensions of exposure as relating to how the immune system is responding. And so if you have a first natural exposure or you're vaccinated, then an immune response may protect you from disease given a second exposure. With sensitization, you might have the first exposure leading to the sensitization and then subsequent exposures triggering an exacerbation. And so in this case, it could be an exacerbation that is related to an asthma flare or a dermal reaction. It just depends on the nature of the allergen.